This presentation provides an overview of our book chapter titled Pathobiology of Intracranial Aneurysms. In this chapter, we discuss IA physiology and pathobiology that highlight specific defects in critical biological pathways that lead to disease. For this presentation, we will focus primarily on insights gained from genetic syndromes that predispose to intracranial aneurysms, genome-wide association studies that have reproducibly implicated specific genes in IA, and lastly, our research that established a new paradigm to define a causal IA gene that is relevant to both familial and sporadic disease. An intracranial aneurysm is a dilatation of cerebral artery, predominantly occurring in the circle villus. IA typically develops in adulthood, and in adults, the frequency of IA is estimated to be 2 to 5%. The rupture of an intracranial aneurysm causes a subarachnoid hemorrhage. For this SAH accounts for 3 to 5% of all strokes and is responsible for 25% of all years lost for stroke victims under the age of 65 due to its earlier median age of incidence and high mortality rate. Our research that established a new paradigm to define epidemiological studies have defined multiple risk factors for IA development and ultimately their rupture, leading to subarachnoid hemorrhage. This presentation IA is more prevalent in women than men, with roughly a 3-to-2 ratio. Although the precise reason for this sex-specific difference remains poorly characterized, it is thought to relate to hormonal differences in smaller arterial wall caliber in women. For this modifiable risk factors include cigarette smoking and hypertension that both lead to increased wall shear stress and induces endothelial cell dysfunction. The strongest predictive factor is a positive family history that is noted in 7 to 20% of patients. For IA rupture, smoking, hypertension, alcohol consumption, and cocaine use have all been implicated. This presentation provides an overview of our book
Genetic factors play a central role in a subset of IA patients. As noted, 7 to 20% of patients have a positive family history of IA. In addition, Mackey and colleagues have performed a twin study where they show that IA occurs almost invariably in identical twins and roughly half of fraternal twins. For SAH, the Nordic twin study reports a SAH heritability estimate of 41%, where the odds ratio for SAH is 51 when more than one family member is affected by SAH.
The American Stroke Association has published their guidelines for intracranial aneurysms.
Of note, first-degree relatives of IA patients are at increased risk regardless of ethnicity. Screening by MRA or CTA is recommended every 5 to 10 years for 3 populations in particular. The first is in cases where someone has 2 or more affected relatives. In addition, patients affected by either autosomal dominant polycystic kidney disease or microcephalic osteodysplastic primordial dwarfism type 2 should be screened. Screening also should be considered for vascular Ehlers-Danlos syndrome type 4 and cartilation of the aorta. Collectively, genetic syndromes with elevated IA incidence account for less than 1% of all IA cases.
In this chapter, we discuss IA physiology and
the genetic syndromes that are characterized by an elevated IA predisposition directly implicate defects in mechanotransduction and disease development.
We will focus primarily
IA occurs in approximately 11% of autosomal dominant polycystic kidney disease patients. This disease is caused by the inheritance of mutations in either polycystin 1, that is abbreviated as PKD1, or polycystin 2 that is abbreviated as PKD2. Both polycystin genes serve as cilia mechanosensors, where there's disruption in renal tubules leads to cellular overproliferation and cyst development. In the cerebrovasculature, the polycystin proteins are expressed in both endothelial cells and smooth muscle cells. The prevailing view is that defects in mechanotransduction in response to blood flow most likely leads to IA.
For this presentation, we will focus primarily
MOPD2 is an extremely rare type of dwarfism where IA incidence may occur in up to 50% of cases. In this autosomal recessive disease, inheritance of 2 defective paracentric genes leads to disease. Paracentrum is required for cilia formation by modulating PKD2 localization. A multitude of genome-wide association studies, or GWAS, have been undertaken to define chromosomal regions and genes implicated in IA across multiple ethnicities. Using these datasets, several meta-analyses have also been performed.
For this
GWAS studies typically interrogate common variants whose minor allele frequency is 5% or higher. Given the prevalence of IA in the adult population, the identified variants in these GWAS have low to modest disease risk.
And lastly,
3 regions in particular have been reproducibly identified in these studies at a high rate. The most highly reproducible region is located on chromosome 9p21.3, and harbors a cyclin-dependent kinase inhibitor 2A and 2B genes, as well as an antisense transcript for cyclin-dependent kinase inhibitor 2B.
Physiology and pathobiology.
The next region maps to chromosome 8q11.23 that contains the transcription factor SOX17.
For this presentation,
lastly, the endothelin receptor on chromosome 4q31.23 has been implicated in multiple studies.
Genome-wide association studies that have reproducibly implicated
the most common region implicated in IA by GWAS is on chromosome 9p21.3 and contains 3 genes that are tightly clustered. This region contains the cyclin-dependent kinase inhibitor 2A and cyclin-dependent kinase inhibitor 2B, as well as cyclin-dependent kinase inhibitor 2B antisense transcript. Each gene has been shown to regulate the cell cycle, and collectively are famous in their roles as tumor suppressors in multiple cancers. Mutations in cyclin-dependent kinase inhibitor 2A are common in cancer and is likely the second most mutated gene after p53. In contrast to cancer research, much less is known about how these genes function mechanistically in IA. GWAS studies have implicated this region also in coronary artery disease and type 2 diabetes. It is currently hypothesized that aberrant cellular proliferation may contribute to IA pathogenesis.
This presentation
Another gene implicated in IA by GWAS is SOX17, a transcription factor that is highly expressed in vascular endothelial cells.
IA physiology.
This gene is required for the maintenance of definitive endoderm in mice. Recently, given the GWAS results, an endothelial-specific conditional mouse knockout has been generated. These mice, when challenged by hypertension, develop intracranial aneurysms with defects in endothelial integrity. Of interest, SOX17 deletion in arterial endothelial cells reduce their proliferation with a concomitant increase in both cyclin-dependent kinase inhibitor 2A and cyclin-dependent kinase inhibitor 2B. This result provides additional support that vascular endothelial cell dysfunction plays a critical role in
pathobiology of intracranial aneurysms.
Another gene implicated in IA by GWAS is SOX17, a transcription factor that is highly expressed in vascular endothelial cells. This gene is required for the maintenance of definitive endoderm in mice. Recently, given the GWAS results, an endothelial-specific conditional mouse knockout has been generated. These mice, when challenged by hypertension, develop intracranial aneurysms with defects in endothelial integrity. Of interest, SOX17 deletion in arterial endothelial cells reduce their proliferation with a concomitant increase in both cyclin-dependent kinase inhibitor 2A and cyclin-dependent kinase inhibitor 2B. This result provides additional support that vascular endothelial cell dysfunction plays a critical role in IA.
That highlight specific
for this section, the last gene that we will discuss is endothelin receptor type A, otherwise known as ENDRA. This gene is implicated by GWAS studies for intracranial aneurysms and coronary artery disease. This gene is a receptor for endothelin that is a vasoconstrictor and has additional roles in cellular proliferation, inflammation, and fibrosis. Endothelin signaling is activated at sites of vascular injury, leading to the idea that abnormal tissue repair may contribute to IA. Mouse knockout of this endothelin receptor causes cardiovascular abnormalities and craniofacial malformations, demonstrating its developmental significance in vivo. Overall, GWAS studies have provided critical insights into IA pathobiology.
For this presentation, we will focus primarily on insights gained from genetic syndromes that
over the past 20 years, we have established a long-term collaboration between the University of Texas Health Science Center at Houston and the Brigham and Women's Hospital in Boston to learn more about IA. Initially, this work was focused on determining if distinct ethnicities had differential incidence of familial IA and understanding if ethnic backgrounds modify risk factors for IA rupture. More recently, we've initiated several studies using informative IA families and whole exome sequencing to identify rare genetic variants that highlight specific defects in critical. We have also established a neuroscience research repository with more than 900 samples from IA patients to do large-scale case control studies. Lastly, our lab uses animal models, cell culture studies, biochemistry, and molecular biology
tools to interrogate putative IA genes to establish their disease relevance. To define a causal IA gene that is relevant to both familial and sporadic disease.
First, we sought to determine if the frequency of familial IA cases varies as a function of ethnicity. Over a 2-year period, we enrolled 200 IA patients from primarily 3 ethnic groups: Caucasian, African American, and Hispanic. For each ethnic group, roughly 20% of IA patients were characterized by having a family history, where the range was 19.4% to 21.6% dependent on ethnicity. This rate is similar to the upper end of the range found in other studies. Lower familial estimates may reflect differences in some ethnicities or healthcare systems.
To define a causal IA gene.
Next, we sought to determine if there are ethnic variations in susceptibility to SAH risk factors such as smoking, hypertension, and alcohol consumption. In this meta-analysis, we analyzed 17 case controls and 10 cohort studies that met inclusion criteria. Overall, the risk ratios for current smoking, hypertension, and alcohol consumption of 150 grams per week were roughly 3.2, 3.1, and 2.5 respectively. The odds ratio for SAH with respect to current smoking was 3.8 in an interrogated population containing multiple ethnic groups and 2.5 in monoethnic ones. This result suggests that certain ethnicities in the multi-ethnic populations likely have increased susceptibility to SAH due to smoking.
This presentation provides an overview of our book chapter titled Pathobiology of
Technological advances, both GWAS and familial studies have become increasingly affordable. Earlier, we discussed how GWAS studies have identified common genetic variants in several gene regions implicated in IA with low to moderate risk. In contrast, familial studies are well suited to define rare genetic variants that have high disease risk. As sequencing costs continue to fall, familial studies are transitioning from linkage studies to whole exome and whole genome sequencing to identify variants that segregate with disease. Unfortunately, the small number of affected individuals within a particular family often leads to the identification of many candidate genes. Moreover, there is very little overlap between candidate IA genes across distinct families that would merit rigorous validation using animal models. As an example, the familial intracranial aneurysm study investigators published their whole exome sequencing results for 7 families. For each family, 4 to 5 affected family members were sequenced, resulting in the identification of 68 rare exonic variants in total, and 2 to 25 candidates per family. Strikingly, no candidate IA gene was identified in more than one family.
Over the course of time, we have established one of the largest sample biobanks for IA patients in the world. Through these efforts, we have recruited many large IA-affected families. Here we will discuss one such large IA-affected family that we termed the CVM family. For this presentation, we obtained informed consent and DNA samples for 35 family members. This pedigree is consistent with autosomal dominant inheritance. In this pedigree schematic, affected individuals are denoted by black fill, while unaffected by white fill. Gray fill represents individuals of unknown IA status. This family has 9 affected individuals where 4 are male and 5 are female. Overall, there are 6 family members with IA and 3 more that experience SAH. The large number of affected individuals in this family provides considerable statistical power to identify the causative mutation that is linked to disease. For this presentation, we will focus
linkage analysis was performed for 35 members of the CVM family. We interrogated more than 200,000 single nucleotide polymorphisms in each individual to define specific chromosomal regions are linked to IA. A LOD score is a statistical measure of the likelihood that a chromosomal region is linked to disease. A LOD score of 3 means that the odds are 1,000 to 1 in favor of linkage. Our linkage analysis in this family identified a single genome region of 7 centimorgan size spanning 13q14 to 13q21. This region had a LOD score of 4.56 or roughly 35,000 to 1 odds in favor of linkage. This region contains roughly 60 genes.
Genome-wide association
to define the specific mutation that causes disease in this family, we employed whole exome sequencing as well as targeted genome sequencing. Given the high penetrance of disease in this family and the apparent pattern of autosomal dominant inheritance,
we focus on rare variants that impair protein coding capacity. Specifically, we interrogated missense, nonsense, frame shift, and insertion deletion mutations in protein coding genes. We discovered a single co-segregating rare nonsense variant in the THSD1 gene that was present in all 9 known affected individuals with IA or SAH and was absent in all 13 unaffected. Each affected individual is heterozygous for the nonsense mutation. This mutation is absent in greater than 250,000 chromosomes in the nomad database and will result in a severely truncated protein missing its C-terminal half. This gene maps to 13q14.3 under our linkage
provides an overview of our book chapter titled
To determine if rare variants were associated with IA more broadly, we performed a large-scale case control study. We analyzed the THSD1 coding sequence in 507 IA probands from our patient cohort, representing both familial and sporadic disease. This analysis resulted in the identification of 7 rare missense variants in 8 patients. Of these, 3 THSD1 missense mutations were found among 162 SAH victims. These patient-identified rare variants were statistically enriched in comparison to controls, and as will be shown later, each variant led to THSD1 dysfunction.
Overview of our book chapter titled
THSD1 is an abbreviation for the thrombospondin type 1 domain-containing 1
physiology and pathophysiology.
This gene encodes a poorly characterized
specific
protein.
defects in critical
When we first discovered the
pathways
nonsense
that lead
mutation in
to
the
disease.
CVM family,
For this
there were very few
presentation,
scientific
we'll focus
reports
primarily
describing
on
this
insights
gene,
gained from
where its
genetic
characterization
syndromes
was
that
largely
predispose to
restricted to its
intracranial
potential role as a tumor
genome-wide
suppressor.
association studies that reproducibly
This gene encodes
implicated
a single-pass
specific genes in
transmembrane
IA.
protein with an
And
N-terminal
lastly,
extracellular
our
domain
research has
and
established a new
C-terminal
paradigm
intracellular domain.
to define a
In
causal
general, the
IA
missense
gene that's
mutations
relevant
affected
to both
evolutionarily
familial and
conserved
sporadic
amino
disease.
acids across mammals, and in some cases, evolutionarily
This
conserved across
presentation
a
provides
range of
an overview
vertebrates,
of our
including
book
fish and
chapter
birds, suggesting
Pathobiology
functional
of Intracranial
significance.
Aneurysms.
2
In this
probands
chapter,
have
we
the
discuss
L5F
IA
variant
physiology
that maps to the
and
N-terminal signal
pathobiology
sequence,
that
while
highlight
the rest of the
specific
missense
defects in
variants
critical
identified in
biological
IA
pathways
patients
that lead to
mapped to the
disease.
intercellular region.
For this presentation, we will focus primarily on insights gained from genetic syndromes
To determine
that
experimentally if
predispose to
THSD1
intracranial
loss of function results in
genome-wide
vascular
association
phenotypes
studies
in
that
vivo,
reproducibly
we used
implicated
2 model
specific
animal
genes in
systems.
IA.
In the
And
first, we
lastly,
took advantage
our
of
research
zebrafish.
established a new
Zebrafish
paradigm
embryos develop
to define a
externally
causal
and are
IA
largely
gene
transparent
relevant
that
to both
facilitate
familial
screening for
and
vascular
sporadic
phenotypes.
disease.
Moreover, developmental loss of function studies
This
can be
presentation
easily performed by
provides an
using
overview of our
antisense
book
knockdown
chapter titled
oligonucleotides
Pathobiology
directly
of Intracranial
injected into
Aneurysms.
fertilized eggs.
In this chapter,
This
we
method
discuss
allows for
IA
the rapid screening of a
physiology
large
and
number of genes to
pathobiology
determine
that
if their
highlight
loss
specific
affects the
defects in
embryonic
critical
vasculature.
biological pathways that
To
lead
model
disease.
THSD1 loss
For
in
this
mammals, we use a
presentation, we'll
genetically
focus
engineered
primarily
mouse
on insights
knockout for
gained from genetic
THSD1,
syndromes that
and
predispose
use this to
intracranial
determine if
aneurysms.
THSD1 loss
Genome-wide
causes IA
association studies
and
that
SAH.
reproducibly implicated specific genes in IA. And
To
lastly,
assess
our research
THSD1
established
function
a new
during early
paradigm
development in
to define
zebrafish,
a causal
we
IA
injected
gene
into
that's relevant
fertilized
to both
eggs an
familial and
antisense
sporadic disease.
THSD1 morpholino oligonucleotide that blocks
This
endogenous
presentation provides an
THSD1
overview of
expression.
our book chapter titled
THSD1 loss
Pathobiology of
during early
Intracranial
embryogenesis leads to
In
cerebral
this
hemorrhages
chapter,
by
we
48
discuss
hours
IA
post-fertilization.
physiology and
Its frequency
pathobiology
was also
that
correlated
highlight
with the amount of
specific
injected
defects in critical
THSD1
biological
morpholino.
pathways that lead to
This
disease.
result has been
For
independently
this
confirmed by the
presentation,
Ducker's
we'll focus
lab using
primarily on
distinct
insights gained
THSD1
from genetic
morpholino.
syndromes that
Altogether,
predispose to
these
intracranial
results
aneurysms.
confirm that THSD1
Genome-wide
in
association
zebrafish
studies that
is important for
reproducibly
proper
implicated
development of the
specific
early
genes in
brain
IA.
vasculature.
And lastly, our research has established a new paradigm
To interrogate
to define a
THSD1
causal
function in
IA
mammals,
gene that is
we were
relevant
fortunate
to
in
both
Nakayuchi's
familial
lab
and sporadic
had generated
disease.
a THSD1 mouse knockout that they shared with us.
This presentation
This knockout
provides an
bears an
overview of our
insertion of
book
a yellow
chapter
fluorescent protein reporter
Pathobiology of
into
Intracranial
exon 2 of the
Aneurysms.
THSD1
In this
gene.
chapter, we
This
discuss
fluorescent
IA
reporter is expressed with the
physiology and
endogenous
pathobiology
THSD1
that
promoter
highlight
and has its
specific
own
defects in
polyadenylation
critical
signal
biological
that
pathways that
terminates
lead
the
to disease.
transcript,
For
thereby
this
eliminating
presentation, we'll
endogenous
focus primarily on
THSD1
insights
expression
gained
from the
genetic
mutant
syndromes
allele.
that predispose to intracranial aneurysms.
THSD1 was initially reported to be expressed in hematopoietic stem cells and in endothelial cells. In mice, we found that THSD1, as visualized by the fluorescent reporter, was expressed in endothelial cells as determined by CD31 staining and not smooth muscle cells as defined by alpha smooth muscle actin staining. This result implicated endothelial cells as the most likely culprit for the cell type negatively impacted by THSD1 deleterious variants.
Pathways that lead to disease. For this presentation,
in the next slides, we present some of our data on the functional consequence of THSD1 perturbations in humans and mice in relation to IA and SAH. Here we show two representative images of IA in patients with distinct THSD1 mutations. The left panel shows a digital subtraction angiogram of a 9 mm saccular aneurysm in patient 2-9 of the CVM family with the THSD1 R450X nonsense mutation. This aneurysm arises from an arterial branch point of the internal carotid artery and the ophthalmic artery. The right panel shows a magnetic resonance angiogram showing a 30 mm right superior cerebellar artery aneurysm in patient NR0756 with sporadic disease, where the THSD1 proline at position 639 is mutated to leucine.
And lastly, our research has established a new paradigm
to interrogate if THSD1 loss of function directly causes IA. We analyzed our genetically engineered mice at 1 year of age. Due to the small size of the mouse brain vasculature, we used a yellow microfilm resin to make brain vascular casts.
In this chapter,
while no intracranial aneurysms were observed in age-matched wild-type control mice, IA was observed in both THSD1 heterozygous and null mice, irrespective of sex. Heterozygous mice have one good THSD1 allele and one defective THSD1 loss of function allele. Marked by black arrows in this figure, intracranial aneurysms in 3 distinct THSD1 heterozygous mice are shown. The top left panel shows a right ICA fusiform aneurysm, while the bottom left panel shows an IPCA aneurysm. Right panel shows a left PCOM aneurysm. These images differ in their magnification. This result implicates THSD1 haploinsufficiency as well as complete loss as a cause for IA.
That highlight specific
in our mouse colony, we notice that a small percentage, approximately 3%, of THSD1 mutant mice died by 4 weeks of age. Gross examination and autopsy revealed massive cerebral bleeding in the SAH pattern. To more formally look at SAH incidence, we sacrificed at 4 months of age 5 mutant mice, specifically 2 THSD1 heterozygous and 3 nulls, as well as 3 wild-type littermate controls. Strikingly, SAH, as shown in this figure, and ventricular enlargement data not shown, was observed in 1 of 2 heterozygous mice and 1 of 3 null mice. SAH was not observed in wild type. Altogether, these results demonstrate that THSD1 haploinsufficiency, as well as complete loss, leads to IA and SAH in males. In mice, it is important to note that these mice were not hypertensive, nor obviously experienced modifiable risk factors such as smoking and alcohol consumption. These THSD1 mice are the first example of a mouse strain that naturally develops IA and SAH without induction by extrinsic factors. After establishing THSD1 mutations as playing a causal role in IA and SAH in mammals, we sought to determine how THSD1 contributes molecularly to IA pathobiology. We performed THSD1 loss of function experiments in human vascular endothelial cells to determine genes and pathways directly regulated by THSD1. For this experiment, we used distinct small interfering RNAs to knock down endogenous THSD1 expression as well as native control siRNAs. 2 control and 2 THSD1 targeting siRNAs were transfected individually so we could define bona fide THSD1 regulated genes and exclude off-target effects. Total RNA was isolated 36 hours post-transfection. RNA-seq was performed, and the global transcriptome was analyzed bioinformatically.
That highlight specific defects in critical
The
biological
bioinformatic analysis of our global transcriptome profiling identified several THSD1 regulated pathways. Strikingly, 4 of the top affected pathways are all intimately connected to focal adhesion kinase signaling, also known as FAC signaling.
And lastly,
FAC is an integral component of focal adhesions and the critical mediator signaling through this macromolecular complex. Specifically, multiple extracellular cytokines, as well as endocrine signal through FAC, whereas downstream targets are additional signaling pathways that include SRC, RHO, and PI3K, AKT, and mTOR signaling.
In addition, TGF-beta signaling is impacted by THSD1 that lead to disease. Although the precise reason for this dysregulation remains unknown, THSD1 contains an extracellular TSP1 domain. In other proteins, this domain has been shown to bind TGF-beta as well as beta integrins. And lastly, our research has established a new paradigm.
This slide provides a visual representation of fact-mediated signaling. A subset of extracellular growth factors and integrin signaling is mediated through FACT. In turn, FACT propagates downstream signal events through multiple pathways that regulate cell proliferation, migration, and survival. FACT also is critical for actin cytoskeleton organization.
For this
slide shows the domain structure of the THSD1 protein, providing insights into function. This protein is a single-pass transmembrane protein. In the extreme N-terminus, there is a signal sequence that is important for targeting THSD1 to the plasma membrane. As to be shown in the next slide, the L5F variant identified in 2 IA probands that maps to this area abolishes THSD1 protein expression. The extracellular region contains an IgG-like domain that is followed by a thrombosbondin type 1 domain. This thrombosbondin domain is thought to bind both TGF-beta and beta integrins. This is consistent with our bioinformatic data that both TGF-beta and integrin signaling is defective upon THSD1 loss. Next, there is a transmembrane region that is followed by the intercellular region. Our data shows that THSD1 interacts with the intercellular protein talon as an integral component of focal adhesions. The nonsense mutation and the 6 IA-identified patient missense variants in this region each led to defective THSD1 function in human vascular endothelial cells.
Titled Pathobiology of Intracranial Aneurysms.
To interrogate THSD1 functionally, we first expressed each IA patient-identified variant to determine their effect on protein expression in human vascular endothelial cells. For this experiment, we used siRNA to knock down endogenous THSD1 expression and then introduced THSD1 constructs that were specifically engineered to be siRNA resistant. These constructs encoded either wild-type THSD1 or each IA patient-identified variant. In the left panel, we performed Western analysis using an antibody that is specific to the C-terminus of the THSD1. As expected, our siRNA did in fact knock down endogenous THSD1 expression. Most variant constructs had similar expression to wild-type or were characterized by a modest reduction in protein expression. There are 2 notable exceptions. The first was the L5F variant that maps to the extreme N-terminal signal sequence that abolished protein expression. The second is the R450X nonsense variant that lacks its C-terminal half. As such, this truncated protein is missing the region that the antibody binds. In the right panel, we show using an N-terminal specific THSD1 antibody that the nonsense variant does in fact produce a truncated protein.
In this chapter, we discuss IA physiology.
Since our bioinformatic analysis showed that integrin signaling is defective upon THSD1 knockdown, we hypothesized that each THSD1 variant may lead to integrins having defective function. To test this idea, we interrogated the ability of human vascular endothelial cells to attach to plates coated with collagen 1, a binding partner for alpha-1 beta-1 or alpha-2 beta-1 integrin heterodimers. Specifically, we showed that THSD1 loss by siRNA-mediated knockdown led to defects in attachment to collagen 1 that can be rescued by expressing wild-type THSD1 construct that had been engineered to be siRNA resistant. In addition, we tested each IA patient-identified variant using similar siRNA-resistant constructs. Expression of no THSD1 variant rescued the attachment phenotype, demonstrating that each variant perturbs THSD1 protein function.
Genome-wide association studies that have reproducibly implicated specific genes in IA.
We sought to understand how THSD1 regulates focal adhesion numbers, as well as signaling mediated through focal adhesion kinase known as FACT, that when phosphorylated becomes activated to mediate downstream signaling.
This presentation provides an
THSD1
overview
knockdown in human vascular endothelial cells reduced the number of axillin puncti
that serves as a marker for focal adhesion. In addition, THSD1 loss decreased the number of phosphorylate FAC puncti, indicating that FAC signaling was attenuated upon THSD1 loss. In this figure, panel A shows representative images, while B and C show the quantification of paxillin and phosphorylated FAC puncti, respectively.
And lastly, our research that established a new paradigm.
A prior study on cell spreading included mass spectrometry to identify protein interactors with talon, a key component of focal adhesions that links vascular endothelial cells to the extracellular matrix.
Pathobiology.
Among the list of putative talon interactors was THSD1. Talon is involved in regulating integrin conformation, integrin signaling, and facilitates integrin collagen interactions.
For this presentation, we will focus primarily on
given talon's role in focal adhesions and regulating integrin. An intriguing hypothesis is that THSD1 and talon interact. As shown in this figure, co-immunopurification experiments demonstrate that this interaction exists, providing a physical link between THSD1 and focal adhesions. Of note, IA patient-identified THSD1 variants, as shown in panel B, were impaired in their ability to interact with talon, highlighting protein dysfunction.
In this chapter, we discuss IA physiology and pathobiology.
Altogether, our molecular data is summarized in the schematic.
Our
model is that THSD1 promotes the ability of talon to interact with integrins, leading to the initial assembly of the nascent focal adhesion. As the focal adhesion matures, THSD1 is displaced or is recycled to function again. When THSD1 is functionally mutated, there is a deficiency in nascent focal adhesion assembly that leads to reduced focal adhesion numbers and negatively impacts FAC-mediated signaling. This signaling regulates the strength of the attachment to the extracellular matrix, as well as downstream signaling events that control the intracellular actin cytoskeleton architecture. These defects lead to vascular endothelial cell dysfunction that causes intracranial aneurysms. In conclusion, deleterious rare variants in THSD1 cause intracranial aneurysms by impairing vascular endothelial cell function. THSD1 protein interacts with talon, a component of the focal adhesion complex, and THSD1 promotes nascent focal adhesion assembly.
This presentation
THSD1 loss and IA patient-identified rare variants reduce focal adhesion numbers, perturb signaling mediated through focal adhesion kinase, and impairs cellular attachment to the extracellular matrix. Our future research is designed to molecularly dissect THSD1 functions and explore in greater detail its role in disease. In addition, we seek to identify additional intracranial aneurysm genes.
We would like to thank our research team and collaborators at UT Health in Houston and Harvard Medical School. The experimental research in the Kim lab was largely conducted by Drs. Teresa Santiago-Sim, Naomi Fang, Yang Rui, and Zhen Zhu. Of note, the Kim lab has long-standing collaborations with Diana Milowitz and John Hagen. In addition, we are thankful for help from John and Christine Simon, Stephen DePalma, and Susan DeMecki at Harvard Medical School.
For this presentation, we will focus
the research in John Hagen's laboratory is currently supported by an R1 grant from the National Cancer Institute, and in the past by GlaxoSmithKline, performing cancer therapy research.
And lastly, our research that established
the Dong Kim Lab has received 2 grants from the National Institute of Neurological Disorders and Stroke, and additional grant support from the Vivian L. Smith Foundation for Neurologic Research. Dr. Kim is also scientific advisor for Taliabri MX Incorporated and XCAP Incorporated.
IA physiology